What are endophytes? Endophytic microbes are hidden within tissues of plants. Endophytes do not cause disease symptoms, instead they are symbiotic with the plant and often beneficial. Plants harbor bacteria within growing cells. These are particularly notable in plant hairs on roots. Kadushi cactus, Subpilocerius repandus in Bonaire. Kadushi fruits. Seeds. Kadushi seedling, bacteria in root hairs. Bacteria in root hairs showing recently divided pairs. All plants naturally host a community of endophytes. But you need a microscope to see them. What about crop plants? All crops harbor endophytes. Bacterial endophytes colonize seeds. Micrococcus luteus is a bacterial endophyte from tomato seedlings. This photo shows tetrads of this bacterium. Tomato seedling root tip showing high ethylene areas to either side of the root tip meristem. Close-up of Micrococcus tetrads in periplasmic space of root meristematic cells. Plant microbes are 1. Soil microbes. 2. Seed vectored. Rhizophagy. Do plant roots consume bacteria to obtain nutrients? Grass roots show numerous roots tip meristems. These root tip meristems are the sites of internalization of microbes and extraction of nutrients from microbes in the rhizophagy cycle. Bacteria entering root epidermal cells in the zone on intracellular colonization at the root tip meristem. A cloud of bacteria is seen around the root tip meristem where intracellular colonization is occurring. The blue stain is aniline blue. Phragmites root stained with diaminobenzidine DAB to visualize reactive oxygen around bacterial protoplasts. Reactive oxygen is visualizable as brown or red coloration around bacteria. The reactive oxygen is the result of superoxide produced by NADPH oxidases on the root cell plasma membranes. The reactive oxygen extracts nutrients from the bacteria that are symbiotic with phragmites. Confocal microscopy. Pseudomonas sp tagged with M-cherry and inoculated into clover plants. Bacteria fluoresce red in the root cap cells. Blue equals chalcofluor white. Green equals cyto13. Red equals M-cherry tagged bacteria. Bacterial protoplasts in periplasmic space. Reactive oxygen defense response of the root cells involves membrane-bound NADPH oxidase. Bacterium Bacillus subtilis. Inside root cells superoxide strips cell walls off of the microbes. Rhizophagy cycle microbes modulate development of seedlings. Microbes trigger the gravitropic response in roots. Microbes trigger root hair elongation. Microbes increase root branching. Microbes increase root and shoot elongation. Bermuda grass seedling root in agarose without microbes showing absence of root hairs. Bermuda grass root containing Pseudomonas. Root of endophyte colonization of root at root tip and re-entry to rhizosphere from root hairs. Bermuda grass seedling root containing Pseudomonas endophyte. All brown spots in roots are intracellular bacteria. Pseudomonas sp in root hairs of Bermuda grass seedling. Bacterial protoplasts shown in hairs. Why is root hair growth linked to presence of intracellular bacteria? Experiments to test the microbial stimulated cell growth hypothesis. What stimulates the plant root hairs to elongate? Ethylene hypothesis. Microbial ethylene stimulates root cell growth. Plant grows in pits and crevices of limestone or in sand along high-salt Caribbean shore environments. Root hair showing microbes circulating along interior of hair in periplasmic streaming. Constant periplasmic streaming of microbes enables efficient nutrient exchange between microbe and root cells and reduces exposure to superoxide. Root hair stained to show microbes in periplasmic space of hair. Clusters of replicating bacteria within periplasmic space of root hair of sedge Fimbristilus cymosa. The red coloration around clusters of bacterial protoplasts is indicative of reactive oxygen secreted by the root cell plasma membrane to induce nutrient leakage from bacteria. Plants increase the numbers of microbe protoplasts prior to releasing microbes back into the soil. Root hair of sedge Fimbristilus cymosa. Periplasmic streaming was measured to move microbes at a rate of 8 to 11 micrometers or second in root hairs of the sedge Fimbristilus cymosa. Microbes circulating along length of root hair. This constant circulation may be a way to induce replication in the microbe protoplasts. Root hair of sedge showing expulsion of bacteria from the soft-walled hair tip. Red staining bacterial protoplasts are seen in root hair. A wave of expansion of the hair protoplast propagates from base to tip of hair, and this wave forces microbes through pores that form in the hair tip. Sequence of periodic buildup and ejections of bacteria from root hairs. Red bacteria are active in antioxidant nitrogen secretion, while blue bacteria are active in nitrogen fixation. This ejection of microbes occurs rapidly with a wave of expansion in the hair cell that begins in the hair base and moves to the tip. This forces microbes through pores in the hair tips. Nutrient absorption function of the rhizophagy cycle. Nutrient analysis in winter wheat with and without endophytic bacteria. Bacteria isolated from winter wheat seedlings. Bacteria removed from seeds by surface disinfection of seeds. Seeds with and without bacteria germinated in seedlings grown in sterile potting mix. Shoots and roots of wheat dried, weighed, triturated and sent for macronutrient and macronutrient analysis. No bacterial control. Bacillus sp treatment. Nitrogen. Nitrogen content in seedlings with and without rhizophagy bacteria/5. Phosphorus. Phosphorus content in seedlings with and without rhizophagy bacteria/5. Potassium. Potassium content in seedlings with and without rhizophagy bacteria/5. Nitrogen-fixing endophytes in leaves of plants. Nitrogen fixation in plant cells involves the following two chemical interactions between bacterial endophytes and plant cells. These interactions between bacteria and plant cells has been termed the nutrient exchange trap, where plants exchange photosynthate for nitrogen from bacteria. Chemical interaction 1. Chemical interaction 2. Table: histochemical analyses of bacterial endophytes in leaves and fruits of plants. Nuclear symbiosis. Host nucleus with bacteria emerging. Stained with nitro-blue tetrazolium to show superoxide around bacteria. In this symbiosis, bacteria in nuclei are provided with sugars to fuel nitrogen fixation. Then as bacteria are released into the cytoplasm, bacteria are hit with superoxide, resulting in nitrogen secretion by bacteria. Tree of Heaven. Leaves of tree of heaven are covered with hairs. Trichomes of tree of heaven. Symbiosis in pitted filamentous trichomes of tree of heaven. Hemp leaves bear trichomes that contain endophytic bacteria. Hemp glandular trichomes unstained. Hemp NBT stains showing bacterial rods around trichome cells. Blue color indicates presence of superoxide. Hemp glandular trichome. Model for how glandular trichomes work. Cannabinoids and terpenoids as oxygen scavengers to reduce oxygen around bacteria. Removal of oxygen is needed because oxygen is an inhibitor of nitrogen fixation. Hops photo showing inflorescences composed of whorls of bracts that bear trichomes. Hops stained with nitro-blue tetrazolium to show superoxide around bacteria. Hops glandular trichome stained for nitrate. Conclusion or hypotheses. 1. In the rhizophagy cycle, all plants internalize soil microbes into their root tissues. 2. In the rhizophagy cycle, plants obtain some nutrients through oxidative extraction from intracellular microbes. 3. All plants obtain nitrogen from nitrogen-fixing bacteria within their cells using the nutrient exchange trap mechanism.